Well, dear students, myself Sarshad Hussain, uh, we started the discussion of locomotor organelles in case of protozoan, and in the previous lesson we have discussed, in the previous lecture you can say we have discussed the pseudopodia, and then we have discussed the flagella. Today we shall discuss the cilia, third locomotor organelles in case of the protozoan. This is the picture of paramecium I have shown you in order to show the cilia present over the body. These hair-like features present on the body are called the cilia. Cilia are exactly similar in structure as that of flagella. So if you remember, the flagella was having a structure made up of the two central microfibrils, then a ring of peripheral microfibrils. Then the two central one were enclosed in the inner sheath. Then there was an outer sheath. Then there were radial spoke coming from the peripheral microfibrils and the arms also. And then the part of the cilia present below the plasma membrane within the cell was called the basal body. So cilia is again having a similar structure. What is the difference between the cilia and flagella? I would like to discuss with you. Number one, they have different method of working. Though both are locomotor organelles, but cilia work differently as compared to the flagella. And this method of working shall be discussed later on while showing you the locomotion by amoeba, cilia, by pseudopodia, by flagella, by cilia, so on and so forth. At this point of time, we are discussing what is cilia. So one difference is that they differ from flagella in the method of working. Other is, in primitive form, cilia covers the entire body, but in more specialized form. Specialized form means the evolved form. Those organisms which are evolved now, they have restricted cilia to certain region of the body only. Third difference, the cilia are generally shorter. You have seen that in this picture. They are shorter. They are not as long as that the flagella was. Flagella was very long whip-like structure, but they are very shorter but numerous. Flagella were only 1 to 8 in numbers, but here you can see they are numerous on the board. Uh, this is the picture which is showing you some infraciliary system. Infraciliary system means this was the cilia that was present outside the body. We have discussed and we have seen in the paramecium, the previous picture. But here you can see that each cilia, after entering into the cell, it has a basal body exactly as we have discussed earlier, as it was present in the flagella. Basal body is also called as kinetosome. This kinetosome just bend toward the right and give rise to a long thread-like structure called the kinetodesma. Now, if you look at the cilia, they are arranged in a row. This is the one cilia, then in the similar row, another cilia is present over there. Or you can see in this picture, this is the anterior portion of the body, this is the posterior portion of body. This is one cilia, this is two cilia, this is three cilia, and four cilia. So cilia are present in row. This is one row, this is another row, this is another row. Here also you can see this is one row, this is another row, then another row, then another row. So what I mean to say over here is that a row of kinetosomes with the kinetodesmata. काइनेटोसोम मीन्स बेसल बॉडी, सो बेसल बॉडी की भी रो है। सीलिया के नीचे हर सीलिया के नीचे एक काइनेटोसोम है, तो इसकी भी एक रो बन जाएगी। लाइक हियर यू कैन सी दिस इज वन, दिस इज टू, दिस इज थ्री, दिस इज फोर, दे आर इन रो along with this kinetodesma. These are the kinetodesma. Together they form this row, form a single longitudinal unit called the kinete. इस एक रो में ये पूरा यूनिट बनता है जिसको हम काइनेटी कहते हैं। ऑल काइनेटिया ऑफ एन एनिमल कंस्टिट्यूट एन इन्फ्लासेटरी सिस्टम, और यही काइनेटी, दैट मीन्स सिंपली द काइनेटोडेस्मा एंड द काइनेटोसोम, they together called as infraciliary system. अगर सीलिया बॉडी के ऊपर से रिमूव भी कर दिए जाएं, ये इंफ्रासिलरी सिस्टम सारे का सारा रिटेन रहता है। ओके, in cilia kineti are not connected, whereas in flagella they are maybe connected. So you can see this kinetia, say for example this is the one row, this is another row, this is another row. One row, in a one row the cilia, the basal body of the cilia and the kinetodesma, they constitute a kinete. This kinete is not connected with this. This kinete is not connected with this, but in flagella you may see that uh, the kinetia may be connected. So this is the fourth difference, that the kinete of the cilia are not connected to one another, whereas in the flagella they may be connected. Then the fifth difference is the cilia, the infraciliature, that is kinetosome and the rhizoblast, is not joined with the nucleus, nor do cilia bears any mastigonemes. We have seen in the flagella that along the long whip of the flagella, small hair-like features were present, which were called the mastigonemes, but here in the cilia they are not present, and moreover, the cilia don't join with the nucleus with the help of the rhizoblast. Then the sixth difference is uh, the cells may divide in two ways, because you know the protozoans, they divide by binary fission, by multiple fission, in which one cell may divide into two. So if you just look at the paramecium, it can divide in two ways, either longitudinally, in this way, that one half of the paramecium may be a mirror image of the other half, or it may divide transversely, dividing upper part separate and lower part separate. Now, keeping in view the position of the cilia and the arrangement of cilia, either you may have the perikinetal division or you may have the interkinetal division. In perikinetal division, the the cilia are cut across all kinetia. That means, say for example, if if this is the row of the cilia, if you cut across like this, so transverse cut if given to the cell of the paramecium, what will happen, that the kinetia may get cut. This kinete, this was a one kinete, this was another, this was another. They may get, all the kinete may be separated, upper half on one side and lower half on one side. So this type of division is called as perikinetal, or also called as homothetigenic division. Or it may divide longitudinally in interkinetal fashion. That means it may divide like this. It may divide from here. In this way, no kinete will get broken into two parts, but left side of kinete may be get separated from the right side, and this type of cleavage is called as interkinetal cleavage, and the division is called as symmetrigenic divisions. In this case, symmetrigenic divisions, the cells are not duplicates of one another, but they are the mirror image of one another. Now, in addition to this, sometimes these cilias they may form few other organelles also, few other structures also. Like in this case, this is a structure of the verticilla. If you look at, so many cilia are joined together, forming a membrane-like structure called the membranal band. Like you can see over here, this this is the this is one band of the membranal. So membranal band means when so many cilia join together, they may form a membrane-like structure. So this is one of the example that cilia may form certain other structure also, like membranils in case of the verticilla. Sometimes the cilia may join together and form the undulating membrane. So this undulating membrane may be formed by joining of the cilia on the body. So in that case, they may not get separated in a hair-like fashion. They remain united with one another, forming a undulating type of membrane. This case is present in case of the polyrosoma. Then there is a third case that cilia may fuse together and form the long cirri. Like you can see, these are the caudal cirri. They are formed by the fusion of the cilia. Or you can say the frontal cirri over here. So they are formed by the fusion of the cilia. So cilia may fuse to form the cirri. They may fuse to form the membranils. They may fuse to form the undulating membrane. Then the fourth type of locomotor organelles in case of protozoan is the myonemes. If you carefully look at the euglena body, you can see these fine lines. These lines are basically the myonemes. It is underlined over here. They are the myonemes. They are again the locomotor organelles. These myonemes that I have shown you, they are contractile fibrils. Fibrils means a fiber-like structure. They may be present either in the pellicle or in the ectoplasm. These fibrils may be straight or may form a network. They may be in the form of ridge or groove. Like in the euglena, you can see the one myoneme, then the second myoneme. This line may show a ridge, and the space present between them is maybe a groove. So they may be in this fashion, ridge and groove, or have alternate light and dark substances. So they may have the light and dark substances. That means one myoneme may be separated from other because some portion of it may be darkly stained, have dark color, and other have the light stain. So they may be they can be looked separately. Then they may be these myonemes may be a contractile microfibrils. In larger ciliates, they are always in the contractile microfibrils. Contractile means anything that can contract. So if the fiber is able to contract like the muscles, it is called the myofibrils or the contractile microfibrils. They may be in the form of the microtubules, like in case of trypanosoma. So it means these myonemes may be present in any of the above form. They are primarily organelles for metabololy. So the myonemes are primarily organs of metabololy. Metabololy means anything that can change the shape of the organism. So it means myonemes are basically meant to change the shape of these protozoan, but secondarily they perform the function of the locomotion exactly in similar fashion as our muscle contract and locomote our body from one place to another. This is all about the uh, four types of two types that we have discussed today, that is cilia and the myonemes, and in earlier lecture we have discussed the pseudopodia and the flagella. This is all about the four types of locomotor organelles. present in case of the protozoans.